Okay, then now we start the discussion. Professor Rossi, can you hear me?
Yeah, yeah. Can you hear me?
Yeah,
Yes,
yeah, sure.
Sorry,
we have
but I
seen
don't
it.
know why the camera is not working. But by the way,
That's
if you can
fine.
hear
That's fine.
me, it's fine.
Okay. Yeah.
Thank you very much for this very inspiring presentation. A lot of very promising results. I have too many questions. Hope that we have enough time. Some more technical, some other more general. You have to say, considering the in vivo studies you did, probably miss something about the route of administration. You found then some problem related to protein corona, something that made problem with the recognition of your exosome plus in your complete system or not?
We did not study protein corona specifically. This is something that we are now looking seriously into it. We are trying to pre-absorb proteins to modify the protein corona, but we haven't looked at the protein corona in these studies. Actually, there is protein corona also on exosomes. So this is a very recent discovery, by the way. It's been recently reported that the protein corona on exosomes is also playing a role. But indeed, there will be very different protein corona in the exosomes compared to the nanoparticles, naked nanoparticles. We have not studied this aspect. I'm sure it has an influence. I'm very sure. So yes.
Have you also considered the role of some biological barriers, or can
We
this system?
what we see is that of course the main barrier, the first barrier is the liver. In the liver, we have 1 billion sinusoids that are only 10 microns in diameter, and they're full of microfilaments. So in the liver, what we have is a retention of a very large percentage of our nanoparticles. So we are working in developing a new generation of particles that will be able to escape liver uptake, and we are doing that in two different ways. One is looking at the protein corona, indeed, but many people have already tried that, and the success is not so high with CD47 or with protein corona. The other thing that we want to do, we are looking at, is to use nanoparticles to saturate the Kupffer cells. And this has been proved in a couple of papers that can be very effective in saturating for a short time the Kupffer cells. So I think the main barrier that we have is to bypass the liver. After we bypass the liver, we are very confident that exosomes as delivery vectors will be very effective because in vitro they have more than 80 or 90% selectivity.
Yeah, for sure. This is a really smart, smart way respect to active or passive targeting that I mean can have the other problem. You're using biological stuff so that is recognized by the system by something that is similar.
Actually,
Yeah.
there are reviews. There was a review by Bob Langer in Nature Reviews materials that brought literature evidence showing that you put antibodies on top of your particles, is more likely that the macrophages will recognize them. So the macrophages are trained to recognize strange things, and you put antibodies, you increase the probability of capturing the liver.
Yeah, and then you are you have also the problem that you're putting too many things onto the surface of nanoparticle, the targets or the antibodies or the prodrug, etc., the catalyst in your way, and you can have the problem that you have mutual interaction between them, and then so the recognition can be difficult. So the use of exosome is very interesting. I'm also interested in this kind of technical stuff about the loading of nanoparticle. Have you tried only with organic nanoparticle, or you think that in principle can be used also? No, you use only inorganic nanoparticle, or can be used also with
For
other
for
kind?
the carbon monoxide reduction method, is this what you mean about
Yeah, yeah,
CO?
yeah, yeah.
This is only is the carbon monoxide is a very mild reductant, so it can only work for novel metals. So we have been able to do that with gold, with platinum, and with palladium, but not with other. If you want transition metal, you have to use other ways. We have created a Prussian blue analog inside an exosome by doing a ship in the bottle approach with chemical components. Okay, but this is a completely different strategy, much more difficult, I would say. But for novel metals, the carbon monoxide works beautifully, so we can reduce it and put it there.
Yeah, but in principle, this ability of exosome to load nanoparticles can be used also with other kind of nanoparticle, so also with organic nanoparticle, is single role with inorganic.
If you are going to use sonication, electroporation, that you can use with any nanoparticle, but they have to be very. The problem is that they have to be certainly under 10 nanometers. Other than
yeah,
that,
very, very small. Yeah, this is the problem.
Okay.
Okay, just a couple of last things. Can you today you talk about cancer. I don't know if you are working also in other field, but in principle, can I think it can be also very interesting with other diseases. I'm talking about the central nervous system diseases when you have a lot of oxidation process and so on.
I agree. This could in principle work for other things. We are looking at infections, so we are looking at delivery to bacteria. So that's very important. But for us, cancer was a big challenge because in cancer you need to be really specific when you deliver things. Okay. So in other scenarios, it's a more widespread targeting. But in cancer, you really need to get to the tumor to be effective. So it's a very interesting application to test how precise you can be in surmounting the value. By the way, if you inject systemically, you're going to have the liver is going to be the same barrier for other diseases, except there's other thing that we are looking at in the case of lung illnesses. We are also using inhaled nanoparticles. So we are making aerosol nanoparticles that can be directly going into the lungs. But for the rest, if you are going to use systemic administration, the liver is the great barrier to overcome.
Yeah, well, just a last curiosity for the future, because I really agree, and also you had very good results about the use of exosome instead of using active targeting, so loading antibody on the body onto the surface. Do you think that this can be a problem then in the manufacturing and obtaining the clinic? So it is something that can to say it be this kind of approach, your approach with exosome is you think is more difficult than for clinical studies, so to continue the next step or not?
No, there's already a lot of clinical trial with exosomes, not only with nanoparticles, but using the native content of the exosome. So there are already exosomes being used in clinical trials as medicines themselves without any loading. Loading. So I don't think using exosomes will be badly received by the pharmaceutical industry, quite on the contrary.
Or even better respect to the other approach. Okay, thank you very much, Professor Santamaria. Very inspiring, and also we study more about that. Thank you very much.
So Dr. Rossi, do you have more questions?
Yeah. Yes.
So
sorry, I don't know how to hand my hand, but go ahead. I will ask later. Sorry.
Yeah, sure. So go ahead. Yeah.
I go. So good morning. Hola, Jesus.
Good morning, Maria. How are you?
So I have a couple of questions. Maybe I missed it, but you treated with your palladium exosomes like primary cancer cells, right? No, immortalized cancer cells, right? My question is, did you try primary cells from cancer patients, for instance?
No, not in that case, but you are right. That would be how to use it. I mean, if this gets into clinic, the ideal way will be to use a biopsy of the patient, grow the cells, and then use the same cells from the same patient, same tumor, exosomes from this cell. This is what we will do. Yeah.
Yeah, because sometimes it really works different in immortalized cells than in primary
Sure,
cells.
I agree.
And also concerning the animal model, did you try PDX model or humanized model or just the xenograph model?
Just xenogram model for now.
Just
We will
xenogram.
use in the new project, we will use PDX.
Okay.
But in that.
My last question, do you think the scale up could be easy with these exosomes and the nanoparticles? And
We are also working on that. We have a proof of concept ERC project in which we are doing two things. One is to scale up the separation of exosomes by tangential flow filtration. So we are increasing the capacity a lot. And the second thing that we are doing is to actually have a very high control on using microfluidic electroporation to actually load them. So we are working on because we fully realize this is a very good question. We fully realize that there's a problem with scale. Yeah, sure.
okay, thank you very much.
Thank you.
Okay. Let's see whether we have more questions from online. More question from online or? Okay, even though I think we are going to stop here. I thanks very much again for everyone for the participation, and I wish you a good life and hopefully see you next time. So, Jenny, we're going to stop here or?
Yes. Thanks everyone once again for joining us today, and we're looking forward to your continued attention for our journals. Have a nice day. We can stop here. Thank you.
Thank you. Thanks a
Thank you.
lot.
Bye.
Thank you. Bye-bye.
Thank
Thank you very
you.
much.
Bye.
Bye bye.
Bye.